assumption that it falls under educational fair use. So, in this, we have a white blood cell, which is a leukocyte. Those white blood cells are not just freely drifting. These leukocytes are able to control their motion by having proteins that interact with each other. They are not just free floating in your bloodstream. They can control their motion a little bit. All these things that we are talking about are just based upon chemical interactions. So there's just chemical interactions that happen with these proteins that allow them to cling, that allow them to make these temporary connections. So this is your cell membrane. Your cell membrane is made of lipids. So this is a lipid raft. It is a hardened area that is free to move around this softened membrane. It's stiffened with cholesterol. It has these proteins that are stretching through the membrane. The parts that are going through the lipid must be able to dissolve in lipids, so they must be nonpolar. The parts that are out in the cytoplasm must be polar because they must dissolve in water. So there's a huge diversity of channels and various molecules that cross the membrane and things that are bound to the membrane. So again, proteins embedded there, the part that's in the membrane must be soluble in fat, the part that's out must be soluble in water. So, within your cells, you have a cytoskeleton. The cytoskeleton is produced from various filaments, actin filaments, microtubules. These filaments are made out of protein subunits. These protein subunits can very quickly assemble themselves and form a lasting fiber. So, they're just repeating subunits of proteins that can link themselves together somewhat akin to how if you made a paper chain. There are then proteins, so this is a severing protein. That is an enzyme that is capable of breaking apart the cytoskeleton. So your cytoskeleton can form, it can be remodeled, it can be taken apart, put back together. So here we have a microtubule. These microtubules are again made of repeating protein subunits that make these hollow straw-like structures. These microtubules are going to form your centrioles. They form the flagella of sperm cells. They form cilia. Again, these microtubules are able to assemble and disassemble. So here we have a vesicle. It's tagged with markers, which is telling the cell what is in it. This is all just being done with basic chemical interaction. So we got a motor protein. This is a protein. It's walking in this funny way just due to chemical interaction between that microtubule and the feet of that protein. It's not thinking, it's just moving according to its own chemistry. So here we have a mitochondria. Mitochondria were their own living organisms. They are essentially bacteria that live inside of all of our cells and provide us energy. So here are the centrioles. These centrioles send out microtubules that are used to help the division of our cells when we undergo meiosis. So this is a nuclear membrane. This is a thin piece of mRNA. This mRNA is going to have a ribosome attached to it. So here we have a ribosome that is attaching. When that ribosome is attached, it is going to whiz along this piece of mRNA. It's going to push out a small polypeptide. So that polypeptide are many amino acids that ribosome are assembling together. Smaller protein subunits will then join up to make larger proteins. So we are going to have these ribosomes. They grab a piece of mRNA, they're going to zip along it, and they are going to make protein. Some of these are going to attach directly to a pore. They can then excrete that protein where it is needed. So it's reading those nucleotides. As it reads those nucleotides, it builds that protein. So you have an endoplasmic reticulum. This is a piece of membrane that can bud out these vesicles. So your endoplasmic reticulum is used in the production of proteins. Those vesicles can then be transported by motor proteins along your cytoskeleton to other parts of your body. This is going to be your Golgi apparatus. Your Golgi apparatus receives vesicles and it sends vesicles out. It is the shipping center of the cell. Again, it is doing these things based on chemical interactions. You have trillions of these little motor proteins moving things about you as you sit and take notes. These vesicles, since they're just made of membrane, they can join with the cell membrane. They can dump their materials out. They can have their proteins join out into the membrane then. So this boundary of fat is what defines our in and out of the cell, what allows cells to maintain controlled chemical reactions within them is they have this boundary, they have a container that they work in. You are a collection of around 200 trillion of these cells that all are working together on basic chemical principles guided by the information in your DNA to perform life's function.